Okay, so to introduce this fantastic yeast to hybrid context, I need to give you a little bit of background as to what the inventor of the process, Jacqui Matthews from the University of Sydney, has in mind for the students. And so basically, it involves the fact that um, GAL4 is a transcription factor that normally controls whether galactose metabolizing genes are expressed. But it has a very interesting structure because it's got a DNA binding domain and it's got a domain which floats around and allows other transcription factors, other bits of machinery to attach so that when they're working, these two domains are working in concert, you get genes switched on. Now, you can actually separate those two domains. And if you do so, of course, then if you've got the DNA binding domain separate from the active domain, then nothing's going to happen. But if you fuse to them two proteins that can interact, you can reconstitute the interaction. So if you put GATA and FOG, which are a pair of proteins that interact, on each of the two portions, then the two portions will actually come together and the transcriptional activity of the GAL4 will be rejuvenated. So this is the principle of the yeast 2 hybrid assay. You're getting two things that you want to test the interaction of to report whether or not they have by showing some transcriptional reporter activity. So what Jacqui uses in the real lab is a yeast strain that doesn't have a GAL4 gene but has the promoter still. It's a really pathetic yeast strain because it needs tryptophan, leucine, and histidine to grow. In order to grow without leucine and tryptophan, you can give these plasmids, which confer to it the ability to grow on tryptophan and leucine. But what these plasmids will also do is they'll also allow the expression of the DNA binding domain and the activation domain. And attached to those, fused to those, you can have the GATA and FOG proteins. So you can actually express, if you express these plasmids, you can express GATA1 mutants and FOG mutants fused to the DNA binding domain, domain and the activation domain, and you can get all sorts of lovely reporting happening because basically if what you've produced on the GATA side fuse it interacts with what you've produced on the FOG side, then the cells will be able to grow without histidine. So all of this strategy is mixing yeast cells with this plasmid and this plasmid, doing the transfection, and then plating out on either plates that don't have leucine and tryptophan, which will grow one or both of these, but equally on plates that also that don't have histidine, and they'll only grow if there's been an interaction between the GATA and the FOG. So it's quite a complicated setup. It's something that Jacqui does in the lab with a limited range of mutants. What she's able to achieve in the simulation is a lot more variation. So if we go and have a look at the way that this works, the student would generate a set of solutions. They've got all their FOG mutants and the empty plasmid here. They've got all the GATA mutants and the empty plasmid here. And they've got some cells and they've got the sperm that's needed just to make everything flow nice and smoothly. So what the students do is they might create, say, 4 tubes, and they then pipette in some of the cells and some of the sperm. So I'm going to put in 100 microliters of the cells into each of these 4 tubes. And you see there's a bit of process happening here. The students have got to basically be able to set up a standard assay. Let's make 1 microliter of, actually I'll make 20 microliters of I think it's meant to be 20 microliters for the sperm. So that's just going to facilitate the transfection process. And then we're going to put in 1 microliter of some of these mutants and 1 microliter of some of these. Now I'm going to make it so that I think we'll just do the FOG9. I think that one works quite well. Let's get a P2 and we're going to put FOG9 into all of these. So every single one of them are going to take up the plasmid that will enable them to make the FOG, that FOG particular mutant number 9. And what we'll do then is put in the empty GBT9 plasmid. Now, I should be changing the pipette, I know, because we'll probably get some contamination now. And then I'm going to put in one of the unknown mutants and another of the unknown mutants. Now, if I close these lids and mix them, and then if I select all the tubes and instead of going through all the stages of a transfection with all the racks at different temperatures and things, we actually decided it was easier just if they pressed a button to say, do the transfection. So you're going to have to wear, if you've made some mistakes with the pipetting, that's going to be, that's going to be reflected in the outputs you get. But there's not really much point in taking students through every single little step of the process. Easier just to go transfect. That's all good, it's done. And now I'm just going to hold down M to make sure they're all mixed up nicely afterwards. And that just helps student angst a bit if you let them get away with that step. There's still plenty of things that can go wrong. Don't worry. So we're going to make some Petri dishes to plate these things out on. So the first ones are the ones which have got just missing the leucine and the tryptophan. So those plates there. And then we'll make some plates that are missing the histidine as well. And we'll just move those into position. And I forgot that I'd actually organized that so that they would do it in a nice order. And now because of that they've messed up slightly, but that's all right. We'll sort them out. So let's pop them all into some sort of nice array. Let's zoom out a little bit. And now I'm going to take these samples and pipette them onto the plates. Now there's 120 microliters in there, isn't there? So I'll put 55 microliters split across the two. So this one here, which was the FOG and the empty GATA plasmid, or sorry, the empty GPT9 plasmid. Again, I should be changing my pipette tip, I know. That's all very naughty, but we're only looking to see what type of readout we get. If I've got some contamination, all the better for illustrating the realism of the outputs that we get. And finally, I'll put sample on here. And now again, in other simulations, we've got a nice oven which we can put these in and do the timing and everything. But we're just going to do incubate plates, see what colonies we get. And we get this lovely array of spots, some blue, some not so blue. The blue ones, of course, are blue because in addition to the histidine genes being switched on in these particular samples, we're also getting the enzyme that converts the X-gal into the blue color. These plates are going to grow everything. They've successfully transfected the two different types of plasmid, but they haven't necessarily produced the right GATA and FOG combination that are going to make the histidine genes express. Notice as well, we've got a little bit of a different size thing happening here. So we've got a number of colonies, we've got a size of colonies. So the subtleties we've got here are really quite amazing. But what's really clever about what Jacqui did was that she's actually made it so that the students can choose which particular GATA mutants they are working with. Now, by default, the 4 mutants that come are these ones here. These amino acid substitutions are the ones in the GATA, and they're the mutants 1 to 4. But the students actually have a massive smorgasbord of mutants that they can choose from. So what Jacqui has done is she's made it so that that you're actually able, in fact, I'll just switch this on so that you can see them in full glory. This is what's provided in the experiment. So the fog mutants and the GATA mutants, and this is the matrix that tells the program how each of those mutants are going to interact. Either 100 means heaps, 90 means quite a lot, 50 means and 0 means not at all. But in addition to doing all of these fog mutants and GATA mutants to start with, Jacqui's got another 40 or 50 GATA mutants, all of which she's come up with an idea of how they're going to interact with the three different fog variants. So you can imagine the amount of combinations that the students have got, either for exploration purposes or for assessment purposes, is incredible. So this is a real tour de force of setting thousands of parameters for interactions and then letting the students explore those interactions by the appearance of colonies on the plates. So absolutely incredible opportunities here for relating amino acid substitutions to functional differences in the binding and actually seeing that experimentally. But of course, you've got to do the experiments right as well. So a brilliant and exciting and innovative concept from Jacqui here.